Good morning everyone. Today's topic is structure of bacteria. A bacteria is a unicellular microscopic organism, which means they are made up of only one type of cell and they cannot be seen using our naked eyes. Bacteria are prokaryotic organism, which means they do not have a well developed nucleus. Instead, they have a structure called nucleoid. This nucleoid is different from a nucleus. Bacteria do not have membrane bound organelles. Now we will move on to the structure of bacteria. This is the structure of bacteria. Bacterial structure can be broadly classified into two categories. One is extracellular structures, the other is intracellular structures. Extracellular structures are the one that are present outside the plasma membrane. This blue color line indicates the plasma membrane. All the structures that are present outside the plasma membrane are extracellular structures. Example, cell wall, capsule, Fimbriae, Pilus and Flagellum. These five structures are present outside the plasma membrane and they are called extracellular structures. Now moving on to the intracellular structure, everything that is present inside the plasma membrane indicates intracellular structure. Example, mRNA, Plasmid, Mesosome, Ribosome, Polysomes, Nucleoid and Cytoplasm. This empty space is called cytoplasm. These are the structures that are present inside the bacterial cell. Now we will see what is a cell envelope. Cell envelope is nothing but the layers that are covering or wrapping the bacterial cell. This cell envelope is made up of two layers in some bacteria. In some bacteria it is made up of three layers. When you look at this bacterial structure, it has three layers, that is three lines are here. No, these three lines represents three layers that is covering the bacterial cell. One is plasma membrane, the other is cell wall, the last one is capsule. Some bacteria will not have this capsule layer. In such cases, those bacterial will have only two layers of cell envelope. First we will see about plasma membrane. Plasma membrane is a layer that is enclosing the bacterial cell. It is the one that controls the movement of molecules inside and outside the bacterial cell. When you look at the structure of plasma membrane, it is made up of a lipid bilayer with membrane proteins. So bi represents two. So two layers of lipid are present. This is one layer of lipid and this is the second layer. These two layers are joined together to make a lipid bilayer. In between these lipid bilayer, we have some certain proteins present. These proteins are present in the plasma membrane, hence it is called membrane proteins. These membrane proteins are of two types. One is integral protein, the other is peripheral protein. Peri means surrounding. So peripheral proteins are the one that is present on the external surface of the plasma membrane. It is called peripheral proteins. They will be loosely attached to the bacterial cell. These integral protein are the one that is integrated along the length of the lipid layer. If see, it is along the length of the entire lipid membrane, hence it is called integrated proteins. And the second topic is cell wall. Cell wall is the rigid layer that is surrounding the plasma membrane. It is present outside the plasma membrane. It is the one that gives the shape to the bacterial cell. It also protects the bacteria from external environment. Based on the composition of cell wall, bacteria can be classified into two categories. One is gram-positive bacteria, the other is gram-negative bacteria. Gram-positive bacteria are the one which has nearly 60 layers of peptidoglycan in its cell wall, whereas gram-negative bacteria will have only 3 to 6 layers of peptidoglycan, which are in turn surrounded by lipopolysaccharide layer. This is the major difference between gram-positive bacteria and gram-negative bacteria. In detail, structure of these we will cover in the next video. And the next topic is capsule. Capsule is the third protective layer that is covering the bacteria. Capsule is made up of 98% of water and 2% of polysaccharide. Since it has more amount of water, it helps the bacteria to resist drying. It also helps the bacteria to escape the process of phagocytosis. Phagocytosis is a process by which WBC will engulf the bacteria and it will digest it. Since it can escape phagocytosis process, it has the ability to cause infection in the host cell. It also, hence this capsule also confers virulence to the bacterial cell. If a bacteria is a capsulated strain, it has the capacity to cause infection in host cell. At the same time, if the bacteria do not have the capsule, they do not harm the host cell. And the next topic is flagellum. Flagellum is singular, flagella is a plural form. Flagella is a whip-like structure that are present on the surface of bacteria. These flagella will help the bacteria in locomotion. Locomotion is nothing but movement of bacteria from one place to another is called locomotion. Why should a bacteria move from one place to another? Bacteria will move in search of nutrients. If the bacteria is a photosynthetic bacteria, example cyanobacteria, they will move towards the sunlight. And if the bacteria detects the presence of any harmful chemicals or toxins, they will move away from these toxic chemicals. For these purposes, bacteria will move from one place to another using flagella. Based on the presence of flagella, bacteria can be classified into monotrichous, lopotrichous, amphitrichous, peritrichous and atrichous. So here one flagella is present in one side of the bacteria, hence it is called monotrichous. If a bunch of flagella are present on one side of bacteria, we call it as lopotrichous. If bunches of flagella are present on both the side of bacteria, we call it as amphitrichous. If flagella are equally distributed along the entire surface of bacteria, we call it as peritrichous, and if the bacteria do not have any flagella, it is referred to as atrichous. And the next topic is pili. Pili is plural, pilus is singular. Pilus is a hair-like structure that are present on the surface of bacteria. Pilus will be larger than a Fimbriae but shorter than a flagella. Pili will be either present on the it will be present either on both the side or it will be equally distributed along the length of the bacteria. This pili will be governed only by the plasmid, P, like this you can remember. And this pilus will be usually 2 to 10 in number in one bacteria. This pilus will help the bacteria in conjugation process. During conjugation process, the tip of the pili will be used to detect the bacteria, that is F-, it is useful in finding an F- bacteria during conjugation. And this pilus will also help the bacteria in attaching to substrates like our teeth, intestine, rocks, etc. This topic is Fimbriae. Fimbriae is singular, Fimbriae is a plural, and Fimbriae is a short, fine, hair-like structure that are present around the bacterial cell. This red color fine line indicates Fimbriae. A bacteria will have nearly thousands of Fimbriae, but pili will be just 2 to 10. So this Fimbriae will help the bacteria in attaching to a substrate. It is so small and it can be seen only under an electron microscope. So far we have studied about the extracellular structures. Now we will move on to intracellular structure. Among the intracellular structure, first we will see about a cytoplasm. Cytoplasm is a gel-like matrix that is present inside the plasma membrane. It has water, enzymes, nutrients, waste and some gases in it. So all the cellular substances are scattered in the cytoplasm. And the next topic is nucleoid. Nucleoid is a region in which most of the genetic material of the bacteria is present. They do not have any membrane. There is no membrane that is covering the, in case of nucleus we will have a membrane that is covering the nucleus, but in case of nucleoid there is no membrane and it is an irregularly shaped region. This nucleoid contains DNA, that is the genetic material, and some RNA and associated proteins in it. This nucleoid is completely different from a normal nucleus. And the next topic is plasmids. Plasmids are small circular double-stranded DNA. They are circular in shape and it is made up of a double-stranded DNA. They have the capacity to replicate on their own. They do not depend on the chromosomal bacteria for their replication. This plasmid is not involved in the normal functioning of the bacterial cell. This plasmid will give some special advantage to the bacteria like antibiotic resistance or resistance to heavy metals. If a plasmid is present, means that bacteria will be a virulent one, and this plasmid can be transferred from one bacteria to another by two ways. One is by binary fission and the other is by conjugation. And the next topic is ribosomes. Ribosomes are the site for protein synthesis. Bacterial ribosomes are 70S in nature, whereas eukaryotic ribosomes are 80S. This 70S ribosome is made up of two subunits. One is 50S subunit, the other is 30S subunit. This 50S subunit is again made up of two ribosomal RNAs. One is 5S rRNA, the other is 23S rRNA. Here this 30S subunit is made up of 16S rRNAs. And these ribosomes are the site where mRNA will be translated into proteins. And the next topic is polysomes. Polysomes are structures which has an mRNA with two or more consecutive ribosomes. So they work on translation process. And these polysomes are, they will be more in number. Always in a cell we will have translation, transcription and replication will be taking place continuously in a cell. And the last topic is mesosomes. Mesosomes are nothing but infoldings of the plasma membrane. This plasma membrane will be infolded in some regions. This is done to increase the surface area of the plasma membrane. So it is a region which is rich in enzymes and it also helps in respiration. And the last topic is mRNA. mRNA is a messenger RNA that is transcribed from the chromosomal DNA. This mRNA will further undergo translation to yield proteins. And we have studied about the entire structure of bacteria. In detail explanation about all these will be given in a separate video. Thank you so much for listening to the entire video. Make sure you subscribe to my channel and thank you for watching till the end.